Yes. Continue. So, Jerry, could you please tell me what do you see on
I
the,
see that slide that you showed first when we tested it. It's up.
okay, and the full
Yeah, full
slide, right?
screen.
Okay.
Yeah.
Okay, perfect. Okay, thank you so much. Yeah, when Charlie first emailed me, and I thought, oh yeah, I haven't done this for a while. So it's actually a good practice for me as well. And this is the first talk ever since, COVID, and since my concussion recovery that I will be giving. So please be patient with me if I stumble on words or maybe repeating things so much. But let's hope that it will be all right, because I talk about things I know, hopefully. So yeah, thank you so much for having me tonight, and thank you for being here. And I know that the times, and this is about dinner times, and it might be awkward for a lot of people, but anyway, thank you so much for being here. So oopsie, what I'm going to talk about today is I would like to kind of give you all a big picture of how cave microorganisms could be of use, or also could be impacted by climate change. And I would lead that into what findings and what kind of research that our lab has been doing, which maybe could help mitigate certain aspects of climate change by using microorganisms. So first, I would like to give a big shout out to a lot of people and a number of fundings that I do not have enough space to put everything in. Even just one project, the white nose syndrome project, we have a whole page of acknowledgement of people names and funding agencies and student names and collaborators. So I would like to give a big shout out to all of my research students, whether they are or they have been or they were undergraduate and graduate levels, and then a lots of collaborators that we have worked with. And obviously the last but not the least at all, you all, like you are cavers, you are citizen scientists whom I and my student have been relying on, and you are all just putting times and resources in without wanting anything back. Thank you so very much. And try to conserve the very fragile habitat that we have. So all of these name is not, I might forget someone, or but basically lots of people had really stepped in and help and provide us samples, or even take us into different caves. So with that said, as I mentioned, I would like to give a big picture of how climate change and how adaptable or how altered microorganisms and their diversity roles and function would be. And then I will go into my research projects, and my lab have done a lot of things. So we will look at whether it could be a reality or it's just my pipe dream, to see whether we could find antimicrobial activity from cave bacteria or not, and where we are at right now. And also another project is we are working on trying to see whether we could find the cave antimicrobial activity from the cave bacteria, and whether that activity would kill multi-drug resistant bacteria, and in the form of biofilm, that which is the normal form when we have infection, instead of thinking about test tube form that we test in the lab. And then the last 5-6 years, our lab had shifted a little bit in terms of working on the drugs from the DARKS project. We went full stream with Dr. Corey Lawson and her team, and many more people as well. Even Martin is actually part of that. So we have developed probiotics for bats, trying to prevent them from white nose syndrome. And besides those projects, I also delve a little bit deeper into what we call as biomineralization. And basically, as the name stated here, bio mean life, right? And mineralization is mean when you have crystal being made from life or living organism. In this case, of course, it's cave bacteria. And why are we interested into this? You might ask. Have you heard of biocement, the word of the kind of biotech industry that we might be able to use enzymes or metabolites that we can get from cave bacteria to look into making certain type of crystal or mineralization for us. And the last one, but not the least, again, this one, I know that it takes so much time and effort and also money to bring one drug out of the cave. And I don't know, even in my lifetime or my working career, I would be able to achieve to do that or not. So we also, our lab, looking at poop sample. Now we instead of playing with the cave sample, now we also playing with poop sample, stool sample to be more academic. But anyway, it's kind of a, my son always said, Mommy, you going to collect poop from the hospital? Yes, but yeah, we do that. So basically this one, we try to find alternative treatment for multi-drug resistant infection that has been a big problem in our society. And I will talk more in detail about that. And if I have enough time left, I would show one quick community outreach that came out from all of this scientific research that we've been doing. But we would be able to make use of scanning electron micrograph from all of these projects to put on an art exhibition. So first, let's talk about microorganism and climate change. Recently, and especially living in Kamloops now and seeing how the summer unfold in front of us in terms of the wildfires and smoke, it hit us really hard, and actually it hit hard before that, but I think this summer is the hardest in terms of the impact of climate change around us. And I still wonder why some of the people would not trust the scientists and would not believe in how climate change had impact a lot of people in the world. But anyway, so mentioning about this, there was a really good paper, and I give the link for you right here. If you want to read the actual article, this one I just screenshot for you to show that there is, and it's not just this group of scientists. There are way more, many other scientists that came out and try to warn us about, the impact of climate change is go beyond what we can see, especially towards microorganisms. And it's actually quite a good read. And I now use this as the best of reading for my student for the International Field School course that I created. We're going to go to Thailand, and that field school course is called Microorganism, Human Behavior, and Climate Change. So that I would take students to Thailand and learn more, show students how to learn more about that. So now let's first have a look at roles and functions of microorganisms in our ecosystems. And as you can see that this picture right here, or this figure that I got out from the paper that I told you about. So if you have a look at this, they didn't even include the subterranean, microorganisms or the ecosystem, right? So they look at the terranean, yeah, and then marine, and then, in basically that's the main thing that often a lot of paper talk about. So I think we really need in the cave microbiology community, we need to really put this out and say that, you know what, it's actually, we need to already link and try to study how climate change impact the microorganisms that lives in the caves. Because, okay, now we, it's kind of, so when you look at the roles and functions of microorganism in our ecosystem, you can see that the microbial primary production, basically microorganisms actually contribute, they fix carbon dioxide, and then through photosynthesis, and then make the primary products for all other living organisms. That one of the major role. Another major role of them, especially fungi or different kind of group of bacteria, they are a big decomposer. The decomposition, process that going on in any habitat, right? And how about the connectivity of the root system from different kind of forests, and whether it be grassland, wetland, mangrove, or wherever. Basically, these are, how should I put it, microorganisms are actually the main pillars of everything. If you really look at molecular level in anything that you look at, it comes down to microorganisms. So with this says, I don't have enough time to even go into how all of this in detail to explain to you. But if you have a look at this picture alone, you can already see that, okay, if there are, weather pattern change, and you can see that the consequences of that is not only to the forest or wildfires, or to the fish in the ocean, or not to the cows in the agriculture and practice that we have, or even permafrost. All of this here, in the tundra area and permafrost or sea ice, it is actually going to expose more of the frozen microorganisms, and let's hope that those frozen microorganisms are not the bad guys. And all of these, when one thing shifts, the balance of all ecosystem actually get impacted very profoundly, and we don't see that until many years from now. So now let's look a little bit deeper how other human activities affect microorganisms as well. So for example, in this figure here, you can see that we add a lot of nitrogen and phosphorus into ecosystem, which actually disturb microbial ecology, meaning that we have done changing or speeding up the evolution of microorganisms in different ecosystem. Or even with the agricultural and industrial emissions here, everything actually affect, because in the air there are microorganisms in the air as well, and then you can see how all of these would affect that. And yeah, when you have a look at this, the way we change the landscape, the way we actually practice husbandry, animal husbandary, or how we actually delete what would be a good, destroy wetlands and make shrimp farm, for example, in Thailand is a big problem with that in Southeast Asian countries. And you can see that they use a lot of mangrove area and wetlands that we have, and then turn that into profitable shrimp factory. And that is just one example. And how about the microbial diversity of the the one that support plant growth. You can see that we actually practice monocultural for so long, and that really changed the microbial diversity of useful organisms turn to be less diverse. And when things happen, it's actually cause more problem to our ecosystem. So now, how about what we are in right now? Global pandemic right here. This is one example of also how climate change exacerbate, pathogens, microbial pathogens. We actually, you remember, right? You all remember that, oh, when COVID-19 first came out, there are lots of people talk about, oh, it came from the bats, the coronavirus and all of that. And that is actually not a false news. It's true, but how come, like when you really look at it, back then, how many years ago on Earth, our habitat and bats' habitat were not really in a gray zone. You know what I mean? Like you don't see a lots of bat back then, unless you really have to go into deep wood or forest or somewhere. But now we are actually, with the population that we have, we encroach into wild animal land and their home. So it's not a surprise to me as a microbiologist to see that now we have a lots of weird viruses coming out of the woodwork. And it used to be estimation on that, oh, we will basically see maybe every 100 years, global pandemic. But no, now I would say, and I don't want to be pessimistic, but it might ring true that we will see new and emerging microbial pathogens coming out of woodwork every 10 years. Okay, so yeah, and a lot of things, like, for example, have you heard of the bleaching of the coral? And I was so sad to hear all the story about that, because basically the temperature change, pH change in the ocean, and then microorganisms that live with marine lives like coral, they just basically cannot live there anymore, and it just became white coral in certain part of the world because the acidity and the higher temperatures in the ocean. So basically coral, talking about why and how coral has harbor a lot of microorganisms in them, it's just like us. It's like us, that we carry coral just like us, in a way that coral also has an immune system through having the microorganism living with them, just like us, human being or animal or plant. We all have microorganisms living in and on us, and they strengthen our immune system. So with this said, I just want to kind of start it off with this in a way that everything is so connected, even the antibiotics, multi-drug resistant bacteria and drug resistant problem that we are having these days. Basically, it came one way or another is related to climate change and human activity. Anyone have a question before I move on to the research? I hope I didn't put like a, pessimistic tone into this talk starting off. Sorry.
I was going to mention, Anne, you could move this to full screen. It's not currently, so.
Oh, it's not currently? I'm sorry.
No, I can see the slides down the side too.
Oh, okay.
That's better. Thanks.
There. Sorry. I thought it's a full screen. Sorry. So here, from that said, we still have hope. We still have hope, and a lot of them too. So I hope that our lab could one day really get to finding or be a part of putting a new antibiotics into the into the society or onto the shelf. So let's have a look at it. At what we have done so far. We have done screening a lot of cave samples in BC and even in New Brunswick and Alberta as well. But the one that really, how should I put it, that we have continued on, because I don't have enough research grant and I don't have enough students that can carry on this work. But so far, we have 3 caves that we really stick to because it had good activities that we wanted to continue. So basically, we have Iron Curtain Cave, and then Helmecken Cave, and then White Rabbit Cave that we have been, trying to continue our work on to. Oopsie, sorry. So and Raspberry Rising Cave as well. I forgot to mention that. But as always, I need more students and more grant to help work to do further study on this one. So this is, for example, what we did with the Raspberry Rising Cave, and I have to stop because I did not have enough students. Actually, now I need master or PhD student to carry this project on. So that's why now it stopped at this preliminary publication. And thank you, Christian Stenner. You see your name in there? Yes. So basically, we found in this study, it started off with Nick Vera, and then we kept on, continuing doing the screening, and look at how beautiful. I would love to go in this cave someday. Look at how beautiful bacterial maps that you can find on the wall there. And what we found is that I would just go through this picture. Basically, this one show how, because with the water and everything to go in, Christian may be able to, explain this better than me. We have to come up with how we're going to put the plates and take it to the cave room that we want to leave the plate in there to let bacteria grow without getting, submerged in the water. But anyway, so the result is that we found this a number of isolates that, especially 2 of the bacteria here, that we found to show antimicrobial activity against multi-drug resistant E. coli, and then the MRSA, Staphylococcus aureus. And there are the other 2 that show activity against non-pathogenic isolate of Staphylococcus aureus. So this one got stopped here because, as I said, I need more master and PhD student to help me take this on. And for Iron Curtain Cave, we have been very lucky with that because I had a number of undergrad students, and some of the students even came from UBC and Chicago, University and Dalhousie, and came to work with me in the summer. So we got quite a lot more done for this Iron Curtain Cave bacteria. So we found, first of all, we found a very thriving diversity of microorganisms that live in Iron Curtain Cave. And that one is the then trainer behind. So don't tell him that I show his behind picture. So sorry, off record. Oh, but we are record. Anyway, so this one is a trip that we went in with the Nature of Things crew. But anyway, so now, so this is his face here. I show you the behind picture first, and then now a front picture. So this cave, maybe Rob can even explain much better, than me in terms of the spectacular feature in there that really grabbed my interest. So instead of just looking at how many bacteria that show antimicrobial activity that could be a new drug lead, we also isolate this, bacteria from soda straw and popcorn. And I will show you in the next few slides to show you how we see bacterial community in there. And it's It's just thriving. So Charlene, you can show your professor and say that it's not a biotic at all. You know, it's just dark. We can't see it, but it doesn't mean that nothing lives there, right? Anyway, so we have adapt protocol and using electron microscope to study them. It used to be so difficult because I have to send all the sample to UBC, and each sample will cost me about $300 to $400 to get someone to prep the sample and then to actually have a look at and give me the image, you know, of who actually live in my in the cave sample that we sent in. So with the work that at TIU, now we have STEM, or what we call as scanning and transmission electron microscope that we have at TIU right now, meaning that I can actually have a fresh sample of spiriothems, like whether it be soda straw or popcorn, put it on the peg, and then put it into this, you know, STEM and have a look at it without having to sputter it with gold and coated the sample with that, because once you shine the electron to it, it will just trigger and then you can't see anything. But anyway, so we got that standardization of the protocol, and now we can absolutely look at the sample ourselves. And this one recently from White Rabbit Cave. Now this one we also need more students to help me to take it to another level. So we just got rejected the paper, but it's okay. Now we actually working on it to submit, to resubmit it, and hopefully they will be okay with it. They just come back and say that it's just preliminary work. They just want to see more. But anyway, it's kind of a, you know, academic career. Sometimes I feel like it's like a game. You have to know how to play the game. Some journal want like really advanced study, but they don't understand a smaller university and smaller labs. We need to also, you know, be able to put some of our preliminary and exploratory, the study out. Anyway, so with Iron Curtain Cave project, sorry that I going back between White Rabbit Cave and Iron Curtain Cave here, but let's continue to the Iron Curtain Cave a little bit more. As I said, that besides trying to find antimicrobial activity that could be a new drug lead from this bacteria, we also have this idea about, you know what, when you really walk into the cave and you see all these beautiful secondary deposits, you know, secondary mineral deposits in any cave. And before, like years ago, there's a thing that, oh, you know what, maybe it's just physical chemical activity that make it happen. But now everywhere we look, it just a lots of microorganisms in there. And you can see that even some popcorns, you know, of each cave popcorn sample, they are not really exactly the same popcorn, you know what I mean? Even though their base mineral are the same and all that. And when you look at them, the microbial diversity and population are totally different. They might have a generic group that they are the same, but there are certain group of microorganisms that are unique to each cave and unique to each of the spiriothem, the secondary mineral deposit that we have. So with that said, I am now so super interested, and today, good news, few months ago, I sent 2 Iron Curtain Cave bacteria that we found to produce crystal in the lab, and we get them to produce it in the lab, in the test tube, but in the plate. And we sent these 2 bacteria to Dalhousie University and got a whole genome sequences, meaning that I want to know all the letter of the DNAs that they have. And I want to what we call as gene mining. We want to mine the genes of these bacteria and see what kind of metabolic pathway, how do these bacteria make and meet, in the term of how do these bacteria make this precipitation of the mineral for us in the cave. How do these bacteria shape the formation of Speleothem sample in different cave for us? So the first paper that came out, this one is I think 2015, but this one we started to look at whole genome sequencing of metabolic study of the cave streptomyces. And this one is a different bacteria that we sent to Dalhousie. So this one, we looked at this ICC1 and ICC2, we gene mine we try to mine the genes that are associated with the drug what you call it, with the antimicrobial production instead of the biomineralization. I will just pass this because basically it's linked to that we try to understand what part of the cave is there any pattern at all, that where should be a place that we pick sample, like is there a place that conducive to harbor more bacteria that produce potential antimicrobial for us or not. And we found out that for volcanic cave, deeper in the cave is actually better place to collect that. So as I said, it's a study of the 2015 metagenomic study. So if I may explain, the classical microbiology study is that when we collect environmental sample, bring it to the lab, and then use a lots of media, different kind of media in the plate, and we plate them out and we study them, and we try to isolate the bacteria that can grow in the lab, right? But it turned out that this kind of classical study yield only or less than 5% of bacteria that are culturable in the lab. So now in the in the microbiological world, we have a new term. We like to coin term, biologists, we like to end the term with something, omic or something, proteomic, metagenomic, something. But anyway, so this one in this case is metagenomic. So this kind of study is giving us more in terms of a big picture of bacterial community and population in a certain environment sample. But it doesn't mean that these bacteria are still alive and viable. But if we found DNA in this sample, so we could kind of interpret the data that this type of bacteria that live in this sample. So it totally the classical study and metagenomic study is complement to each other. So that's why now all the microbiology lab around the world, we combine these two techniques together, because if we use only classical technique, growing them on the plate, we don't get a big picture of it. But the metagenomic study, even though we get big picture, but we can't get the bacteria that alive from it. So this has to be, all together, like to complement each other. So the first set of data that came out, we found more antibiotic producing genes in deeper point of collection in the lava tube, actually, compared to karstic caves. And this is really true about Iron Curtain Cave too, because Iron Curtain Cave is karstic cave. And when I looked at all of the bacteria that show the antimicrobial activity, is actually what you call it, earlier in the trail. It's not deep in the cave. But anyway, we need to do more of that research to confirm whether this observation is true or not. So now when we try to find new drugs, normally when we get infection, bacterial infection, they actually work together you know, they have society, they have community, they communicate to one another. They are just like us, but in a much smaller scale. That's all. So when they infect us, they are not alone. So they actually they grow in the structure of we call as biofilm, meaning that they grow in a layer of different species to make them stronger, because when they have layer upon layer of bacteria and different species at that, antibiotics is hard to penetrate this. So the last few years I also kind of not shifting my technique to find new drug, but shifting, how should I put it, maybe expand. I would say expand and not shifting, because I'm still doing testing the, you know, the type of bacteria that are not forming biofilm to find new drug. But now I also trying to test the cave bacteria on the biofilm of multi-drug resistant bacteria in a hope that we could find something that can kill, you know, the actual community living closer to that type, than the, you know, something that we found in the test tube, basically. So before I move on, anyone would like to ask any question?
Yes,
Or
I
if
have
not,
a question, Anne.
okay, yes.
Yes, so with respect to you mentioned that in Iron Curtain, or sorry, the lava tube cave, it was at greater depth that you were able to identify bacteria which had comparatively more antimicrobial producing genes.
Yes.
And I was curious, do you have a hypothesis about why that might be the case? Now, presumably there's something about the microclimatic conditions that are, you know, selecting for these genes at that area. But I'm wondering, is it depth per se, or is there something, or could it be something else that's idiosyncratic about where you sampled from?
Yeah, you know what? I think it's so hard for us to answer right now, because it's such a huge study. I mean, if we were to be able to give a definite answer to you, because
do you have a hunch of your own?
A hunch, I think it has to do with the micro habitat in each cave, and I don't even know whether each cave would show. That's why we need a more, you know, more data.
So it's not, it can't be, you can't rule out particular idiosyncratic characteristics, and it's not depth per se.
No, and as you all know, right, when you go into each cave, they are so unique. So I can't even say like unique in a lots of things, right?
It'd be interesting. If it's a broader pattern, it'd be interesting, and then that's a hypothesis that could be further tested.
Exactly, because that study we compare Hawaiian lava tube cave and then Azole, you know, so we didn't have enough or bigger data to really definitely say, oh, is it just really a depth in a deeper, you know, a depth and deeper cave that it shows us? Could it be the, you know, base mineral, and could it be the micro habitats of, and how about animals that live in there? It's a lot of things. That's why I just feel we just see that pattern, but I don't think that it could be a definite, you know, saying that depth would be the way to go for lava quite yet.
Presumably it's not depth in and of itself. It's some property of the cave that would be correlated with being at depth, like airflow or the temperature.
Absolutely. And how about the dripping water? Where's the terraneal, you know, like the up, the what you call it, what that bring in, right? Organic matters level and all of that. There are a lots of things. Yeah.
Right.
Mhm.
Thanks. Thank you.
Yeah, but thank you for the question. So now with this biofilm, we actually collaborate with Dr. Jack, University of Montreal, and it's very interesting. Next time I will show more of the pictures, but we are working on it right now. Instead of using the plate, we actually using 96 well plates and grow the biofilm of the bacteria in the wells. Like it, 96, we have 96 wells, and each well come with a peg. So you just put the peg into the nutrient and add the bacteria in and give them some time, and they will grow into like this. Isn't that so cool? So this one is scanning electron micro image, micrograph. This one it show free living bacteria, like single cell living bacteria. Do you all see my cursor? When I point out, can you see the cursor with the slide or no?
Yes.
Okay, so these are like free living pseudomonas. Like pseudomonas is actually a short sausage, if I may say, a short sausage kind of bacteria. So these are them free living, but when we grow them longer, it grow up to like this. Now this one you can see free living and starting to be bacterial biofilm. This one is film, meaning that biofilm, like they are growing on top of each other, layer up on layer. And this is the hardest to kill using antibiotics. So that part, I cannot share the result right now. Tomorrow I will get the report from the student in the lab. So next time, please be patient with me. Next time I will show more of the result from this project. It is very exciting. We actually find something that kill bacteriophage, and we are now trying to test carbinoid as well, the CBD oil. We have a colleague here at TRU who got a big grant and connection with one of the CBD cannabinoid company in Canada, and they want us to test on the biofilm of the bacteria. And we will see more work on that front as well, not only cave metabolites against bacterial against biofilm. So now this is exciting because this summer we gain a lot of results from this biomineralization and the potential that we can use it in the biocement industry. So let's have a look at actually yesterday Keegan, I don't know, Rob, I think you might remember him. Hey Keegan, one of really good student that I had. He's now living in Alberta and going to go to medical school, but he, during work, he put this all together, this study from Iron Curtain Cave, that we got popcorn and soda straw and we isolate bacteria from that. Yesterday, he was presenting this at the 2020 European Mineralogy Conference. Yeah, so here are a few pictures and result that we found. So we did the chemical analysis of it as well, between comparing of the elements, element that we found in popcorn sample and soda straw, which is very interesting, isn't it? Because when you think about it, it's all calcium carbonate. How come other element is so different like this? Like this one, soda straw sample has way more magnesium. And what else? For example, barium or aluminum and all of this. So I'm trying to crack this as well, like why? And as this, going back to the question earlier, that when you ask about, is it just a depth or is it other thing as well? I think there are way more to everything. It's so micro habitat that way. So we have been isolating. So from the popcorn sample, we isolate, my student isolate, like, you know, a number, different number that you can see here, and we sequence them and we identify them to be all these bacteria here. But one of the thing is that you might see the terminology here that we talk about urease candidates. Why? What has the importance of this is that I want to look into the metabolic pathway of this. I want to see how these bacteria make ends meet in a way to actually precipitate calcium around them. And one of the theory long before that has been presented, because it's not only cave bacteria that can precipitate mineral around them. Any bacteria that live everywhere, like elsewhere in the soil, in the dirt, or other you know, other environment, there are a certain groups of bacteria that, you know, typically they precipitate mineral around them. And the theory goes that they have urease enzyme that involved with urea metabolism, and they use that metabolic pathway to help precipitate the, you know, the mineral between them. So we still trying to crack more of this and trying to understand them. And like I said, I'm really happy that today I got email back from Dalhousie saying that the 2 strain of our bacteria are actually done. This one that we sent out, Cyclobacter and Athrobacter, this one that get fully sequences now. So Richenda, I don't know whether Rob, you still remember Richenda? Richenda is now working in San Diego. She got master's degree, but she's still right with me, and she going to, yes, thank you, Rob. Yeah, to look into the genes in this bacteria. So now this is it. Beautiful picture. We got this bacteria to precipitate mineral in the agar plate in the lab. I was like jumping up and down when it happened, because we waited and waited, and it took about 3 to 4 months. I almost lost my patient and say, oh, just throw them away, you know, it's not going to happen anymore. But actually, finally it happened. And COVID helped with that, because if it not for COVID, we might have already been too hot-headed and throw all the plates away, because it's starting to get dry out, you know. So you have to talk to Keegan about that, Rob. Keegan was like, one day call me and say, Anne, I was about, you know, throwing all these out, and when in the process of throwing the other batch out, he saw crystal in the media when he's trying to watch the, yeah, exactly, to watch the test tube. So you can see, I don't know whether you can see much or not. So these are scanning electron micrograph. You can see already that A and B here are from popcorn spiriothym. Popcorn spiriothym, for some reason, and this is a picture of popcorn spiriothym from Iron Curtain Cave. I think it's about physical chemical characteristic of it. Because when you think about dripping water coming in, bacteria can just stay, right? And then eat up organic matter and, you know, precipitate calcium carbonate and all of that. So you can see that there are way more bacteria in this scanning electron micrograph of A and B here compared to C and D. These are soda straw scanning electron micrograph. We can't see much of bacteria at all here, there, and there. And I'm not even sure whether that are bacteria or not. You know, this is a little bit here, and that's about it. But it kind of makes sense, because when the dripping water dripping down, how much bacteria would go wash off with it, right? And then would stay in there. So, but still though, when we isolated them and grow them in the plate, we find a lot of them. Anyway, so these are the crystal-like formation that we observed in specific media. It is quite beautiful. I'm sorry that it's a little bit small here, but yeah, but you can imagine how it, you know, and one of the interesting thing is that different morphology of the crystal are actually previewed to us. So this one is give you a little bit more idea. So these are actually made by iron curtain cave bacteria and made in the broth media. So we started them off to grow in the agar media, and then now we grow them in the liquid media, and they produce crystal too. Look at how beautiful they are. Like this one looks almost like flowers, and this one look almost like, I mean, diamond cut or something like that. And this are just different, so different morphology of them. I am still learning with all of this. I don't know all of this at all, but and then now we know that, okay, if we actually give them yeast extract or more nitrogen source, it induces them to produce more. So we're trying to understand what make them take. That's what we are trying to look into here. And different bacteria producing different shapes of the crystal is so interesting. Any question before I move on to bat project? Okay, and how much time do I have left? Am I actually boring you out of the falling down the tree here? Sorry. Yeah.
No, this is great.
No, it's
No,
good.
this is
Okay.
awesome. Thanks, Anne.
Okay,
Yeah,
good.
yeah.
Thank you. It's weird, hey, when you actually do this and you don't see people and body language and it's so quiet and you are the only one who talk. It's like, yeah. So
Well,
anyway,
Nobody's left, Anne, so I guess you're doing okay. And
okay,
it's very
perfect.
interesting. Continue on. Take as
Perfect.
much time as you need.
Okay, perfect. Thank you so much. So now let's switch gear a little bit. I'm going to talk about bats. And in my life, I just feel, now it's all filled with fault back in 2015. Because, at the invasive species conference on TRU campus, he pulled me over, Phil, and he said, Anne, meet Corey, and you guys can work together. And since then, Corey and I just hit it off and just being, bat biologists and microbiologists working together, trying to find a, way that we can help protect bats, especially Western bat species, from white nose syndrome. So let's see where we are at right now. I cannot, because it's really a 6 years project and it's a lot of things to talk about, and I don't want to like go into too much detail for you because I have other projects to show you too. But basically, I will just give you an update a little bit on what we did and what we have done so far. So basically, first, we started off in 2015. We got funding from NFWF and a friend of BAT from the US government. And the research grant helped us to in the first year, to collect wing swabs from bat species, from Western species, through Corey's team. And they all send the swab to my lab, and then we grow bacteria. We grow the bacteria that live on healthy bat wings onto the plate, and we isolate those, and we got about 2,000, actually, to be exact. I think it's 1375 different strains, 1,375 strains of bacteria that we got after the preliminary screening, that they are not repeating bacteria, they are not the same bacteria. So we got 1,376 of that. And my student and my postdoctoral fellow then came from India, Dr. Somya Ghosh. He actually he and the students screen all these bacteria against PD, against the fungus that kill bats, that cause white nose syndrome in bats. And we got a number of them that show activity to kill the kill the PD, the fungi. So from that, we send them to McMaster, our collaborator, Dr. J.P. Zhu, in his lab. He actually did, he and his Adrian Forsyth, his PhD student at the time, they did what they call as combination test, meaning that they test all of these combination of bacteria that we send to them that show activity in my lab, and then they test them against one another, combine with that dose, they do the antagonistic combination test and see who are working together the best. So we got four different bacteria that work together the best to be a cocktail of probiotics that we now actually have the second year of field trial done right now. So it's actually very promising, activity that we found. And it's going to be the first preventative method that use the Western species, use the bacteria that isolated from Western species of bat. And these are just a fun kind of slide to show you what we have to do. Like, I am a microbiologist and I never thought in my lifetime that I'm going to be working with bats, because in Thailand, when we talk about bats, they are big, they're like chicken, the size of it, so different species. But this one, when Corey text me and say, oh, I have a dead bat, you guys can use, okay, I'm going to send a frozen sample to your lab. And I was thinking that it's a big, big boy, big, what you call it, big bat. But when I opened up the box, it was like tiny little one, cutest thing. But sorry, but it's already dead. But anyway, so I thought, oh, this is not too bad at all. I was kind of scared of bat before, and now I'm not. But anyway, so we work with the Kamloops Wildlife Park. We have to build this structure to mimic the to be what we call as a summer captive trial, because now we work that it tested against in the lab and it works. So now we have to test the probiotic cocktail in the live bat. And what we can do is we can't do anything much, because my lab and TRU are not certificated, that are not certified by animal health care in that way. And Craig Willis, who's actually the big guy in Bat World, he has a lab in, if I remember correctly, in Winnipeg or Regina, I forgot, but in that area. And he said to me and Corey that, and don't use my lab, because he built a walk-in fridge, and he said that you have to have at least $500,000 to do that facility to test this. So Corey and I, we decided that, okay, we would just make our own, summer captive trial. So this is one of them. We make 2 of these at the Kamloops Wildlife Park, and we put the bats in there, and we feed them and observe them, record them, and then in the bat box we put it out, so that we inoculate this with bat box with a probiotic cocktail with bacteria that we hope to help prevent them. And we got really good result from that summer trial. So in the winter of 2019, with just about COVID hit, we had to set up a bat hibernation experiment. And with we don't really have $1 million to build walk-in fridge or make a cave, artificial cave. So I told my student Nick, my master student, to go out and buy a beverage cooler, and just we modified into a cave. And so we put our bat to hibernate in there, and this is in my garage, because TRU didn't want to have anything to do with bats on campus, nor we have a certification to allow them to be on campus in the lab. But anyway, it's all worked out. So this one, we have the humidifier in there to make the humidity to be closer to the cave, which is more than 90%. And we have the the camera and the sensor and everything in there. When bats actually wake up and come to eat mealworm and to drink water, we capture all these video and observe their behavior as well. My student, he totally, like, this younger generation of Canadian, of us, they are just so technology savvy, and he worked so hard to put this all together. Yeah, it wouldn't happen without Nick, my student, my student. So here is the example of the chamber box that we have to go through, because like, oh, this is the most difficult project I ever had in my life, I have to say, in my 25 or so years career of being an academic microbiologist. We didn't know, because we were thinking that, oh, maybe, when we have the bacteria, we would just put a sponge, and then the bat will climb in and then pass through the sponge, and they will get the bacteria through their wing and stuff. No, it didn't happen that way. So we have to even just, think about how we going to apply this to the bat wings. Are we going to spray it in the cave, or are we going to do, how are we going to do this? How are we going to apply this to the bats? Yeah, we have to, everything, it was like from scratch. So this one, you can see that on my right hand side is the experiment that we spray the powder, clay powder with bacteria, coated with bacteria in it. And on my left hand side, you see that we use the brush, the paint brush, and make the clay powder mixed with water and bacteria, and then just paint it on. And it turned out that the powder spray is actually work better for bat wings. So before I move on to another project, is there any question? So this summer, as I mentioned, we are still doing the field trial. This will be our second field trial. Yeah, we have a couple of sites that Corey set up and work with her team, and we get a bigger colony of bats to test, and we move the production lab from UBC to UVic and produce more probiotic bacteria, and we distribute out to the test site. And we should have by early next spring, we should have result coming back. Okay, so now I can look, and I mean, how should I put it? As some of you know, in my other talks before, that to bring one antibiotics out to the shelf, it can take up to like 10 to 25 years and billions of dollars, million of dollars, not billion of billion of dollars. So I try also to find alternative treatment, like instead of using antibiotics or trying to find new antibiotics, what else can we do to help fight against infectious diseases, I mean. So turn out I play with stool sample from the hospital, especially because I test the water sample from different caves, and I never find any bacteriophage. So bacteriophage, as the name states, bacterial came from bacteria, right? And phage are the word that we refer to viruses. So this type of viruses actually can kill bacteria and is host specific to only bacteria. So this is an idea of trying to use biocontrol agent instead of using chemical to help us fight infectious diseases. And the best place that we can find bacteriophage are hospital sewage sample, I found. So this one, it was back in, I forgot, this study is back in maybe 2014. But this summer we got a grant to go further instead of this study. This one, we actually isolate viruses from Kamloops sewage treatment plant. And we found the one that killed multi-drug resistant E. coli that we used. But this summer, we actually got more grant on this project. So I got 3 very, very good students to work on this, to continue this project. So we actually expanded our stool sample. Instead of only from Royal Inland Hospital and Kamloops sewage treatment plant, we went to Vernon, Penticton, Kelowna, Salmon Arms, and other places to screen. And we found a very, very good, different kind of viruses, which too bad I haven't had the report from my student yet. Tomorrow he's going to submit me the report for the result, but I already know that we found a lot of interesting stuff, but I do not have the picture to show you from this summer result. So the last one that I'm going to touch on is that with all of that said, I have a lot in our lab. We have a lot of pictures of bacteria in different format. So we try to do community outreach by combining science and art together. And this has been going on since 2014, and we still have art exhibition from here, from here, there, and different places, especially local school and science center in Kamloops that have our artwork right now. So, so these are, you can see the biofilm of bacteria here living together on a mineral deposit, as you can see on the calcium carbonate on here. And then one of our First Nation students saw that, and she said that it looked like the Chuchwap map, her nation map. So she put that into this picture. So now I think I actually ran out of my voice. I need a little bit of water to, I think let's set open floor to talk about things instead of just having me alone to talk about this. So before I end this and open Q&A floor, I would like to thank my husband Joe and my son Ryder for being my rock and let me go to a lot of caves. Thank you. And thank you everyone for your attentions. And if there are any questions at all, any suggestions, or any philanthropist that send my way, we can talk about this. Let's open the floor to discussion. Thank you.
Thank you, Anne. That was a good presentation, very interesting. So if people have any questions, push your button and ask away.
Yes.
Hi, Dr. Ann. You said earlier you use different medias on your plates. What sort of medias do you use?
Thank you so much, Jason, for asking, because I didn't mention in the talk that cave bacteria. Hi. Oh, it's still light out. Where are you? I like
I'm in Port Alberni.
Oh, Port Alberni, nice. So basically, with the cave bacteria, we learned that we cannot give them a rich media. We cannot give them the same type of bacteria that E. coli can grow on. We actually use what we call it, poor nutrient data. Poor nutrient agar, because when you think about a cave environment, there are not a lot of organic matters in there. They are what we call as oligotrophic, the types of of the environment in there, meaning that this type of bacteria are actually, they do not want a lot of food at all. They can just go by little food. So a lots of media that we use in our lab are R2A, the type of media that marine microbiologists use to grow ocean microorganisms as well. But a lot of the time we would use it 10 times diluted. We don't use normal strength. Like we would dilute 10 or 100 times of the normal strength for that, because otherwise you kill the cave bacteria. They wouldn't grow, and they would grow. They would take a long time to grow, like 2, 3, 4 months. Some of them 6 months. Mhm.
Thank
Yeah.
you.
Are they
You're
more
welcome.
susceptible to contamination? Like, are your antiseptic protocols like even more enhanced than your standard microbiology lab?
Yes, yes.
Yep.
All of my students actually work in the biosafety cabinet when they grow this.
Right. Yeah.
And then we do like UV the plate, on tops of what we buy is a pre-disposable, like pre-sterile disposable plate, right? And sometime, because we know that it's going to take a long time to incubate them, so that are prone to contamination. So we would UV on top of that, UV the plate before we put the sample in. And a lot of the time we would buy what we call as like the deep ditch, like instead of normal size of the petri dish that typical microbiology courses or lab use, we would buy like really deep one. So we can actually give them really thick agar, because we actually incubate them in the incubator for a long time. Yeah, we learn a lot. Like, this is not it's not a typical microbiological technique by any means. Yes. And go back to Jason's question as well, lots of media that we use, we would collect cave environmental sample, especially dirt or soil sample in certain cave. If we have that, we would collect that in bulk and then autoclave them and add those in tap water. And that will be our nutrient for the cave bacteria, because they don't need a lot.
Yeah.
What were the different calcium salt substrates you're using there? I saw there's a few different ones.
Oh, what do you mean? Like all those calcium carbonate, like crystal that we have?
Yeah.
No. Those are actually bacteria made that.
They generated those calcium.
Yes.
Oh, okay.
Yes. If you look back, or if I can share screen, or if I can send this PowerPoint for you as well if you want, and then you can look at the picture again. We have a negative control, meaning that the plate that we didn't put bacteria on, and you wouldn't see anything, you just see a clear agar.
Okay.
But the bacteria, we add calcium carbonate, just normal powder calcium carbonate in the plate. And over time, bacteria produce that and make all those beautiful crystal formation in the plate.
Okay, I wasn't fully sure what was going on there. I'd love to have a copy of it for sure, because
Yeah.
I was trying to read it and listen to you at the same time, and then it'd be
Yeah.
on to the next thing. So yeah, that'd be wonderful.
Yeah, it is very stunning. Like we were blown away, right? Because you know that bacteria are actually, you kind of in what you call it, intuitively know that bacteria are heavily involved in speleothem formation in the cave. But now we want to learn how do they do that. So we add calcium carbonate into the plate to start off for them to grow, and they just make that powder of calcium carbonate as a nutrient into the crystal. Mhm.
What do you know about the formation of moon milk then? Because I know there's been some research done on it, but I'm not up on the latest with that because that's always been a bit of a mystery.
It's not really mystery. I think it's kind of, maybe I should rephrase myself. It's not kind of a mystery in that sense. I think this is it. I think bacteria are actually, when you have calcium carbonate or just calcium in different form, calcium, what do you call that? Calcium charge, right? The element. And bacteria has enzyme or the type of protein that could aggregate that into different form of the crystal. So it would be interesting to look at bacterial population in the pearl, you know, and see whether those bacteria are the same with the bacteria that make soda straw and make popcorn or not.
Mm.
I bet you that maybe some of them might be similar, but some of them would be unique, different kind of bacteria that would, you know, that would have the type of protein or enzyme to aggregate calcium into the pearl. Because when you really think about it, whether it be soda straw, you know, stalactite, stalagmite, what else, all of these spiriothym, they are the same base. I mean, if it's the same chemical elements, why are they in different morphology?
Mhm.
So
But
it
it's-
has to be biological activity help with that, right? Because if the base element is calcium carbonate,
But is moon milk mostly calcium carbonate then too, or is it something
Yes.
slightly different? Oh,
yes.
okay.
Yeah. Yeah, but moon milk in lava tube in Hawaiians, I read one paper on that. They have copper and their moon milk is green color. It
Oh.
has copper leaching into it. Yeah,
Nice.
I know, really nice. But anyway, it's so interesting because if we think that the calcium carbonate or gypsum, you know, calcium sulfate are the base of all of this, a different kind of spiriothym. Why the morphology of it and the formation of it are so distinctively different, and in each case is so different too. So,
Mhm. I noticed with cave popcorn, it seems to be a function of, I don't know if it's proximity to the entrance, but wind. There always seems to be quite a lot of air movement where you get popcorn. So I'm wondering if that's bringing in things that are helping in the growth.
yes, and in terms of microbiological point of view, when you
Yeah.
compare soda straw and popcorn, like the place that bacteria can live happily, popcorn has way more surface, you know, like surface and the way the humidity and wind and all of that, you know, to help sustain that. But soda straw is like you just get the dripping water and dripping. So it must be hard for them to kind of hang on and then do their work, right? Yeah. So
Mhm.
yeah, it is very interesting. I'm still a novice at this. Sorry, I can't answer all the questions yet.
Do you think it's possible that there's ancient microorganisms trapped in the limestone, and as it's dissolved, they may be released? And I mean, it was an organic environment at one time. Do you think that's a possibility?
You mean
Like those
Lily?
organisms are trapped in the limestone, and then as they're precipitated into the cave passage, maybe certain ones have different, organisms in them, which causes different creation of formations, right?
Yes, that could potentially be. And another thing that I'm thinking about is that, you know, if the bacteria, no, I don't know whether I can explain them as the same as that they have conscious, but I think, I don't know, maybe they do have conscious, you know. If they know that when they actually precipitate this around themselves, they are actually going to get trapped. But why are they still doing it? I don't know, maybe I'm thinking too deep or thinking too much or using my human species point of view to, you know, to trying to understand this.
Well, is there some these Isn't there typical of these microorganisms, there's something called horizontal gene transfer? Is there something about it facilitating the environment for more the overall growth? Because typically too, your typical like medium, like bacteria, they grow on each other, and the ones in the center will die out. Isn't
Yes.
that the case?
Yeah,
Right.
it's like sacrificing for better good, right?
Right.
What you call it, for the survival of the species.
Yeah. So it's kind of more like not kin selection is not the right word for bacteria, but it's I think there's a term for it. It's been a while. My molecular biology is fuzzy at this stage. That's
Yeah, no worry. Thank you. And you know what? And now when I look back when I was a university student and now, the terminologies that we use to explain the behavior of microorganisms are different too. Back then, we never used the word behavior, communication, society, community, population. We don't use those terms for bacteria, but now we do use all of this because we know more about them, and we know that actually, in a way, maybe, yeah, there are way more than what we used to think about microorganisms. Mm-hmm.
Yeah, so yeah, I guess, no, that's just me pondering, but yeah, potentially it's the sacrifice for the,
yes.
I guess, enhancing the overall environment, but possibly.
Yeah, it's possibly because,
anyway.
Because everywhere we look, they are fossilized. They are actually trapped in there. Yeah. It's kind of interesting, to kind of ponder about, what's happening, like, yeah, why? Yes. Any other questions? Maybe you guys have suggestion for me. Where should I go to find money? I need more research money so I can hire more students to do this. Yeah.
Yeah, good question. I don't know.
I know government try not to, give me grant when it comes to antibiotics. So
well, they don't want to because they want to sell drugs.
exactly. Thank you,
Yep.
Rob. Yeah.
Hey, Anne, I have a question for you.
Yeah.
Is there a way to date these microorganisms that you
Honey?
find in? Is there a way to date these microorganisms that you find embedded in soda straws or calcite?
If they are fossilized, yes.
Okay.
Actually, there is a lab. One of the cavers, but he passed away. He has a connection with the European. I have to find. Yeah, there is. What his name is Eric Stein. He's a Norwegian or. Yeah,
Stein Erik Lauritzen.
Honey?
Stein Erik Lauritzen.
Yes, that's it. Thank you. That is the one that we send the sample to get dated.
Yeah, he worked with Derek Ford for many years, and he's just continued on. But yeah, they do amazing work for sure.
Yes.
It's Billy Thumbs.
Yes.
Yeah.
I think we could do that, Rob.
Okay.
Yes. Oh, and Rob, I'm trying to figure out the vehicle right now.
Okay.
Yeah, I will text you later about Iron Curtain Cave. We want more sample.
Not a problem.
Thank you.
I should be available anytime after September 30th.
Okay. Mm-hmm.
Yeah.
Thank you. Yeah, and I can
Anytime.
do only weekends though.
Okay, perfect.
Mm-hmm.
I can make it work.
Yeah, thank you. Other questions or any suggestions? And I have to say, my research, and I can never thank you enough, everyone in the caving communities. Thank you so much. Yeah, I can never, my lab and my student can never had such research experience without you all. Thank you so much.
Thank you, Anne.
Thank you, Anne. Great.
Yeah,
great
thank
talk.
you.
Yeah.
Thank you.
Thank you, Anne. So I guess that's the end of the official Anne presentation.
Yes.
I don't know if anybody wants to hang around and talk caver talk or anything else. We have still a little bit of time. So everybody's welcome to stay, and so
And Jerry, I
yes.
can send Jerry and Charlene, I can send my PDF of my PowerPoint into it. Is a big file with all the picture, but I can convert it to PDF, and I can actually either email you or drop in the Dropbox, and everyone can go back and have a look at it if you need to.
Well, that would be great, Anne. Thank you.
Yeah,
Yeah.
and I'd like it too.
Mhm.
Me too.
Okay, I will do that. So I guess I will bid my goodbye right now and go feed my cat. She's been meowing.
Yeah.
It's right clockwork, 7:30 something, she's meowing. Thank you so much,
Thank you,
everyone.
Anne.
Here.
Thanks,
Yes.
Anne.
Great.
Good night.
Bye. Good night.
Okay, so recording.